Both this chapter and the next chapter deal with immunity. They will synthesize a lot of the things you've learned in the earlier parts of this course, and in a much more applied way, sort of talk about the human body, how we respond to disease, which I think is the most interesting to people who are about to pursue a medical path. I like to think of immunity when we talk about it in this course as being sort of two sides of the same question. So from our perspective, what we're trying to do is avoid being food for microbes. Obviously, from the microbes' perspective, they are trying to survive. And again, the immunity sections will really focus primarily on pathogens, and as we know, that isn't the majority of microbes, but it is really important to medicine. So this chapter that we're going to start talking about today and finish on Thursday is innate immunity. That's non-specific, and it's the things in your body that defend against pretty much any pathogen that's coming in, regardless of what specifically it is. And we're going to then follow that up with a discussion of adaptive immunity in Chapter 17, where we talk about how we respond specifically to individual pathogens. That includes detecting their antigens, the things on them that are unique that we can make antibodies to, and then how we maintain that immunity over time. But it's really important to understand that these two branches of the immune system, even though they're talked about as separate, are actually really integrally linked. So they talk to each other. And you'll see that as we talk about innate immunity, we'll have references to adaptive immunity that we haven't even covered yet. Something else to note is that innate immunity relies heavily on pattern recognition. So even though innate immunity is a non-specific response, it's not that it isn't intelligent at all. It is detecting things about pathogens in general, but typically it'll, for instance, detect things that are common to many bacteria. So it's sort of able to respond to a broad array of pathogens without knowing which specific species they are. Okay, so we're going to need to remember a couple of things from the chapter we just covered in order to really appreciate this section of the course, and that has to do with pathogenicity and also portals of entry. So just remember that when we talk about pathogenicity, which is how pathogens make us sick, that it was an important component was how pathogens adhere to their host target cells, how they're recognized or how they avoid being recognized, and certain kinds of toxins, either endotoxins or exotoxins, as well as this idea that pathogens can mutate, and therefore if we've developed a specific response to them, we then have to start over and make a new one. And also I want to remind you of the three main ways that pathogens gain access to us, right? So through the skin, if we've actually got a wound or we are dealing with something that actually makes holes in skin, which is very rare. Mucous membranes, which are vulnerable, obviously, and the parenteral route, which is what? What was the parenteral route of entry? Yeah, so it's like breaking the skin. You have to cut it or somehow puncture it. So there's just, this overview again, this is like a sort of artificial compartmentalization of immunity, but it's helpful in terms of being able to sort of remember everything, to talk about it in these terms, the first line, second line, and third line of defense. So the first and second lines of defense will what we be focusing on in Chapter 16. And the way that these are sort of organized, the best way to think about them is from the outside in. So your first line of defense is going to be the outside of your body, and then if the second line is going to be sort of non-specific things in the body, and then the third line of defense is innate or adaptive immunity. So in the first line of defense, we have, of course, the very most important thing that's protecting us is what? Our skin. That's a great barrier to pathogens. Mucous membranes and their secretions are also really, really important. They are the first contact that we have with many pathogens, and therefore we do have a lot of defenses there. And this first line of defense includes our normal microbiota, because where are these found? Where's our normal bacteria that we just coexist with? It's found on the skin and mucous membranes, right? So both of those places, we've got our own physical barrier, we have the things we make, then we've got these organisms living with us that are actually helpful. The second line of defense is all of the things that we do or have that protect us once the pathogen's already made it past that first line. Okay, so they got in through the wall of the castle, and now they're running around inside the rooms, and how are we going to protect ourselves? That includes a lot of the white blood cells, various kinds of different white blood cells, many of which will find, capture, and engulf to digest microbes and other dangerous things. We also use inflammation being a local thing that's happening where a lot of white blood cells and chemicals are coming to one site where, for instance, if you get a paper cut and it turns red, that's inflammation. Fever, which is a sort of systemic experience, and antimicrobial substances, things that we put into our bloodstream and our lymph fluid in order to protect us. So again, the second line of defense is if we've already had a breach in the wall and now we've got to do something about it, but it's not specific. And when we go into chapter 17, which we will talk about next week and possibly on Thursday if we have time, we'll talk about these specialized cells that have an ability to recognize and specifically deal with a pathogen, that's a particular bacterial strain or a specific virus. So our first line of defense is our physical barriers, and they also include antimicrobial substances that we'll put into our mucous membranes and the normal microbiota. So this is sort of like a really simplistic view of a human, right? If we were just thinking about it sort of topologically, and we've got this, very strong skin defense, and then these somewhat more porous mucous membranes, and this is what we look like if we just sort of think about ourselves functionally. If we look at the skin, we know that it's got lots of layers, right? And the other thing about it is that the top layers, which are dead, have a lot of keratin, which is waterproof. So we've got, we're wearing a raincoat, basically. And there are many layers of tightly packed cells. The pH of the skin typically is a little bit acid. And why would that be helpful, to have an acid pH on our skin? Yeah, a lot of bacteria can't grow in low pH conditions. Now, some of them can, and we'll sort of talk about that in a second, but if we look at what our skin is primarily composed of, we've got this really thick barrier that is water repellent and also typically is dry compared to mucous membranes. So why is being dry helpful? Cells need water, right? And our skin's always coming off, which helps us because how is that helpful? Then if there's something on there, it'll potentially be scrubbed off on a regular basis, just our cells will drop off. And so we can sort of get rid of infections to some extent because that's happening. Now if we look at the mucous membranes are a different situation. So here's our skin again. This is just a schematic of the skin with the many, many layers. These are all nuclei, by the way, these blobs. And they're sitting on a basement membrane, which is just a lot of protein secreted by this bottom layer of living cells. That's just a way for them to sit down on something. And then a bunch of connective tissue, which is also made of proteins, and then the rest of the body. Now if you compare that to the mucous membrane, say, for instance, this cell, does anyone know what kind these are? These are representative of both. This is sort of what the gut cells look like. Also, these kind of are what the respiratory system cells look like. So they'll typically be sort of tall and skinny, but there's only one layer as opposed to many layers. So this is a more vulnerable barrier. And they're wet, right? They're typically moist, which means, again, they're a little bit more vulnerable. Now, some of them still have a pH protection, right? Any come to mind? Your stomach has a very low pH. That's primarily for digestion, but nonetheless it does help us because there are many pathogens that, for instance, make us sick if they get into our skin, but don't survive ingestion because they're killed by the acid in the stomach. Also, the mucous membranes of the reproductive system in women especially tend to be a little bit acid, so that's helpful. Now, one thing that these mucous membranes do is they have, you'll see intermittently these goblet cells, they're producing mucus. That's to keep this this tissue moist. But it also, they'll have cilia on top, and in the respiratory system in particular, this is really helpful because the mucus actually traps pathogens that are trying to get in. And then these cilia will push the mucus up, out of your respiratory tract, outside, and expel things. So if there's something here, it kind of is an irritant, and that triggers the cilia to push things out. Which is why if you get something in your, if you inhale something, you have this like compulsive need to cough it out. But it's also working all the time, even when that's not happening. So there may be mechanisms for some of these mucous membranes to move things. One of these is called the ciliary escalator, and this is what moves microbes out of your lungs. So even when you're not actively coughing, this is happening all the time. Those cilia are there in a big ooze of mucus, and if something gets inhaled that shouldn't be there, it just moves this all out towards the front of your body to get rid of it. Your gut also goes through these kinds of movements to move things out of your body in the other way, obviously. And then there's other mucous membranes that just have a lot of fluid, and so they tend to, by gravity, things just flow out. And that's how these mucous membranes are kept clean. Lastly, there's something called the lacrimal apparatus, which is producing the fluid that goes across your eye, and that cleans your eye on a regular basis so that you can, get dust and microbes out of there. So this is just a picture of the ciliary escalator in your lungs. And what you can see is, in this diagram, you can see that there's this mucus-producing goblet cell that's making all this mucus to trap anything that shouldn't be there, and then these cilia, which are working together to push this in one direction. And in this pseudocolored electron micrograph, you can see a whole, it looks really a lot like an undersea landscape. You can see all these cilia and these trap bacteria, and they're going to be moving it in the direction of coming out of your body. Now, one kind of, if you've ever had surgery, has anyone here had surgery where they had to go under? Do you remember, or did they use the torture breathing device when you come out of anesthesia and you have to do the little reps with the thing where you blow into it? They're trying to get your respiratory system to remember how to do this, and also to get things moving. Because what happens when you go under anesthetic is that those cilia in your respiratory tract go to sleep. And so anything that gets in there is going to just go deeper and deeper, and there's nothing to stop it. Okay. So one of the risks, one of the major risks of going under anesthesia in the hospital? That's the result of that. Getting a pneumonia, right? So there are other things that can happen too. If you've been intubated, mechanically ventilated, you're more susceptible to certain kinds of opportunistic infections that other people don't get. So this is just a case study that's included in the notes. You can read about it on your own, but I think that's really interesting. Now, if we look at the lacrimal apparatus, this is a series of structures in your eye that secrete fluid so that your eye gets washed out regularly. It's like having your own saline wash. And what you can see is that the glands are here, and this whole part of your eye is designed to wash that fluid across your eye regularly every time you blink, and then drain it out this duct. Okay, so that's your skin and mucous membranes. Now there are also antimicrobial substances in your fluids. So in sebum, which is the sort of waxy stuff on your skin, there's something that's a fatty acid that is fungicide. So it prevents funguses from growing on your skin. Without that, you would experience a lot more skin fungus. Your skin's also really salty, right? Because when you sweat, there's a lot of salt on your skin. And anybody from the organisms that we've talked about or worked with in this lab know which ones are commonly found on skin that are in fact able to survive this salty environment? Staphylococcuses, right? Because remember when we did that MSA plate? What did we put on there? We put Staphylococcus aureus and the other one, Staphylococcus epidermidis. And we could tell the difference, right? Because one of them ferments mannitol, and that one tends to be more likely to be a pathogen, which is why it's useful to know. But Staphylococcus epidermidis is a relative that's very benign and also is a halophile, right? It survives on your skin. So a lot of the microbes that are normal, like the normal microbes on your skin, are going to be halophiles or able, at least halotolerant. They're going to be able to survive these salty, low acid, or low pH conditions. So as I said, your skin has a low pH between 3 and 5. Your stomach has a pretty low pH. And also some of the other fluids in your body are low pH fluids. And that's protective. The other thing that we have is lysozyme. That's found in almost all of these fluids. So the sweat, your saliva, and urine all have lysozyme. And I don't know if this is ringing any bells for you, but lysozyme is the same enzyme that bacteriophage use to drill into bacteria, to get in, right, to inject their DNA genome. And it's the same thing they also use to explode the cell on their way out. So it is an antibacterial enzyme. There's also things like peroxidase and transferrin in saliva. So peroxidases are going to protect us from oxygen species that are toxic. Transferrin binds iron. Now, I don't know if you'll remember this from our discussion of chapter 15, but we talked about something called siderophores. Do you remember what those were and what they did, or why they were helpful? Siderophores. They scavenge iron. And iron is important in the electron transport chain. All of those electron carriers in the membrane have iron that's part of their enzymatic mechanism. And we can't survive without iron. Microbes are similar, they can't survive without it. So we are in a war with our pathogens in particular, where they're trying to extract iron and we're trying to get it back. And by stealing it back from them using transferrin, we can try to prevent them from being successful. So this is just a diagram to summarize the first line defenses in a typical person. So we've got all sorts of antimicrobial substances in our saliva. We've got also antimicrobial substances in tears, and we have the respiratory system, which has what to get particles out, what physical. It has that ciliary escalator, right? So the escalator coming up through them all. We've also got, the skin itself. We have the mucus and various kinds of cilia on our mucous membranes. We have pH. Low pHs in a lot of different parts that help us to kill things that are pathogenic or discourage them from taking up space. And we also have normal microbes that we haven't talked about. So what do these do? I talked about this in the very beginning, and I've emphasized it throughout this course, that the normal microbes that coexist with us are important for excluding pathogens. So they sit in places on cells where pathogens would like to sit, and they just basically take up space and prevent the pathogens from colonizing. They consume nutrients, so pathogens would go hungry, and they alter the environment. So they secrete wastes, and many of these wastes are antimicrobial compounds because they're in a war with other microbes. And they might have made some sort of truce with other members of the community that normally lives with your body, but the pathogens have not entered into that truce, so they are still going to be attacked. And they also can often make toxins. Now there are commensal microbes where, and the definition of commensal in biology is that one organism benefits and the other one is unharmed. So these in our bodies can sometimes be opportunistic pathogens if they get into the wrong place, right? So for instance, E. coli in the gut, which is a normal and helpful microbe there, if it gets into other parts of your body, in particular in your urinary tract, then that will cause a problem. And it's important, and we know this, right, that if you take a course of antibiotics to deal with some infection, you can have a secondary consequence, which is what? You're swallowing these antibiotics, which hopefully you're doing because it's necessary to fight back something that's already taken hold. But then what happens? Yeah, you're depleting your good bacteria in your gut in particular, which leaves you vulnerable until that regrows or it's replaced. Leaves you vulnerable for a secondary infection or for opportunistic pathogens to take hold, in particular something like Clostridium difficile. So here's just a little It's not comprehensive by any means, but a sort of idea of the different species of microbes that are found in different parts of the human body typically. So there are some of these bacteria that will discourage a lot of other microbes because they secrete fatty acids. E. coli also synthesizes colicins that discourage a lot of pathogens. We've got lactobacillus in women in the vagina create a low pH, and other organisms have these kinds of, these are just some examples, but there are lots of our normal microbes that sort of create the environment that's actually good for us. And as I've already mentioned, antibiotics can disrupt this. And what you see over here is just a list of some of the major species. It's not comprehensive by any means, but where things are found. And some of these you'll recognize because we've studied them, like E. coli, and various Clostridium species, and various Enterococcuses, and Streptococcuses, and Staphylococcuses. A lot of the skin and mucous membranes are populated with gram-positive cocci, and there's a lot of gram-negatives in the gut. And this is not a 100% breakdown, but it is somewhat a general summary of the situation. So now let's move on to our second line of defense. And remember, what's happened now is something has gotten into the body despite the fact that we have a really strong skin barrier, despite the fact that we've got stuff in our mucous membranes protecting us. And, you know, it's either gotten past the ciliary escalator or the lacrimal apparatus or in through the skin somehow. So these second lines of defense are cells of the immune system that are found in circulation in the blood and the lymph fluid. We also have this whole process of inflammation. It's like a big coordinated effort. We can experience fever, and there are antimicrobial substances that we'll talk about inside the body. So first, let's talk about the immune system cells. They're circulating through your body in the blood. They're increased during infections, and the way we make them is a process called hematopoiesis. Does anyone know where this takes place? Where new blood cells are generated? Regularly, all the time? It's in the bone marrow, right? And so in the bone marrow, there is a hematopoietic stem cell. That's like the great granddaddy of any kind of white or red blood cell that you might need. And it has the ability to differentiate into all of these different things. Okay, so all of these different kinds of terminally differentiated cells that have very specific functions go through these different lineages. And so, for instance, you could make red blood cells, right, that are not immune cells. They're just there to carry oxygen. You can make platelets that are important for clotting. You can make various numbers of white blood cells, and these are all the kinds that have specific functions. So some of them make antibodies. Some of them are part of our adaptive immune response that we'll talk about that verify the antibody-making cells. We've got various kinds of macrophages that engulf things. They're all slightly different in terms of what they do. So some of them, they're all made in the bone marrow. Some of them mature while they're circulating. Others go to different tissues to mature. Others stay in the bone marrow. Okay, so we'll just briefly go through them so that you can kind of understand what they do. And this part of the class we will revisit when we do our final lab, Lab 10. And we're going to do a fun activity. Well, it's fun for some people. Depends on your personality. But if you like counting things and doing statistics and calculating percentages and just it's very satisfying, then you will enjoy this. Otherwise, you'll find it boring. I like it, but that's me. So there's different kinds of white blood cells that you can find in your bloodstream, and depending on the levels of them, that can indicate different kinds of infections. So we will actually count those in one of the coming labs. But in any case, we'll just briefly go through what you might see if you were looking just at a typical blood smear. So you're definitely going to see lots and lots and lots of red blood cells, and their function is what? Carry oxygen and transport carbon dioxide. We've also got these myriad of white blood cells. So we have neutrophils that are important for phagocytosis, meaning what? What does that mean? That means they run around finding stuff and they engulf it and then digest it. There's some of them, like basophils, secrete histamines, and that word is probably familiar to you in what context? Allergies, right? So if you have an allergic response, you're most likely going to take an antihistamine to try to combat what these guys are doing. But they're there for a reason. The histamine is part of an inflammation response. It's just in an allergic situation, there's a higher level of inflammation that is necessary to keep you alive. We've also got some eosinophils. These guys are important for killing parasites. So this means some cells are running around engulfing microbes, but you can imagine that some things are too big to engulf, right? So can you think of an example of a pathogen that wouldn't be small enough to just take into a cell and chop up? What about a tapeworm? Or even some of the other like smaller parasites. Protozoans that are parasites might be too big to engulf, right? We can engulf bacteria and viruses, no problem, but if you can't do that, then these eosinophils will stand on the outside and they will attack using various techniques that we'll cover in order to kill the parasite from the outside. There are also macrophages and dendritic cells, both of which are involved in phagocytosis. There's something called natural killer cells, and we're going to come back to these. They are considered technically part of innate immunity, so a non-specific response, but it's going to be most helpful to understand what they do when we go through chapter 17 and come back to them, because they're going to sort of fill in the gap of something their adaptive immune response can't do. T cells and B cells are definitely considered part of the adaptive immune response, which we'll cover in chapter 17. T cells are our cell-mediated immunity. B cells are humoral immunity. I'll cover that in chapter 17. And they mostly produce antibodies. And then, of course, we've got platelets. What you'll do in the lab next week, I think, or maybe it's Thursday, I can't remember. When we do lab 10, you're going to look at all of these different types of very common white blood cells and count them up to see whether they're in the normal range or not, and then you'll be able to tell if the blood smear that you're looking at is from a sick patient and what kind of sickness they might have. So I think we're doing this on Thursday, but I don't remember. I'll have to check my schedule. I'm going to skip over this part and just say a little bit more about phagocytes. So the word phagocytosis comes from phago, which is Greek for eat, and cyte, which means cell. And it's literally a cell whose job it is to find things and engulf them. And what you can see here is the macrophage. It's kind of got this really lumpy external surface, and it's sending out these scary-looking projections to kind of grab onto these microbes. And we'll talk a little bit more about how it knows what to grab onto, because there is a process. But it sort of grabs onto them and starts bringing them in like a tractor beam, and then actually consumes them and digests them inside of the cell. So the problem with phagocytes is that if you turn them on, they can be a little bit crazy. And so they may not just engulf and damage the cells that you want them to. They can damage tissues as well, and so they're typically running around inactivated. You have to turn them on. And they get turned on by T cells, which are part of our adaptive immune response that we'll talk about. So this is one of many examples where there's a cooperation between the innate immune response, which we're talking about right now, and the adaptive immune response. They're also activated by some kinds of chemical signals. Now, if a given macrophage can't destroy what it's trying to destroy, then many of them can get together to form a giant cell, and this forms a granuloma that isolates organisms that can't be destroyed. And there's one famous example of this, which is tuberculosis. Now, remember, we talked about this when we talked about staining. Mycobacterium tuberculosis and other species in that genus have an interesting cell wall. Does anyone remember what's special about the cell wall of Mycobacteria? It's waxy. And that mycolic acid, which is the waxy lipid, is part of a mechanism that protects them from being digested. So even though they can be detected and brought into a phagocyte, they can't be digested. And so then the phagocyte is just left there with this undigested cell inside of it, and it doesn't know what to do. So it talks to all its friends that also have these undigested cells, and they form this giant community to kind of just at least try to quarantine the problem, even though they can't really get rid of it. And so over time, these big masses of infected cells can actually destroy tissue. And these granulomas appear in people's tissues, not just in the lungs, which is where they first appear, but in your muscle, in your genitals, all over your body and your bone, as TB gets more and more severe. Because if you can't get rid of the microbes, then sometimes they'll escape from this big community that you've put them in, right? This big, enclave, and they go out and make more infection, and then they get engulfed by more, and they form more granulomas. So it's quite a difficult disease to eradicate for that reason. Another kind of phagocyte are neutrophils. They're very powerful, but they only have a very short lifespan, which is useful. Since they're more aggressive and they could do more damage, why would we want to have them be fairly short-lived? We'd like to have them turned on when we need them and then just go away. Like, we don't want to have lots of them roving around just doing damage willy-nilly. And they have this ability to throw out a trap, basically, that contains a bunch of, it's basically like an extracellular net to grab onto things. So they're pretty powerful. It's kind of crazy what they can do. So once you have found a target and you've brought it into you, whatever kind of phagocyte you are, because there's different ones, what happens is basically first you are engulfed by endocytosis, and that little round particle that's made, this compartment, fuses with a lysosome. So here's the phagosome, which is just when you engulfed it with and surrounded it with a membrane, you made this little membrane-bound compartment, which is the phagosome. Then it gets fused with a lysosome. Now, who remembers what lysosomes are? Remember we talked about all the different things that you could find inside of a eukaryotic cell? Do you remember what lysosomes were? Everyone was always forgetting that they exist at all. What was their job? You're close. They have all the digestive enzymes, so they're like the stomach of the cell, right? So we have this lysosome stomach, but what it is just like this. It's a membrane-bound compartment that contains digestive enzymes. Here's a membrane-bound compartment, so we just fuse those two together, and all these digestive enzymes get put with the microbe that you're trying to get rid of. And now this is called a phagolysosome, and it goes about digesting whatever it is. And this leaves eventually sort of this partially digested gunky whatever, which is called a residual body, that goes to the surface of the cell and sort of empties its contents out, right? But by that point, hopefully you've destroyed whatever this is, unless one of these steps is blocked. So if you're a microbe and you're at war with the humans, then there are different places where you can try to fight back. Now, if we're mycobacteria, where are we fighting back? Right here, right? We have this mycolic acid cell wall that makes it hard for these digestive enzymes to work. There are other microbes that will prevent different steps. So there, for instance, are some that never get engulfed. And I've talked about a component of the bacterial structure that can be protective, that can prevent the initial step of being engulfed at all. Does anyone remember what that is? You can do special staining to find it. It's not a flagellum. It's not an endospore. It's a capsule. And you'll remember that I've told you that certain species of bacteria give you pneumonia, and their sibling species that can't make the capsule don't. They're not infectious. They don't give us a disease. And it's because the one with the capsule doesn't get detected by this, you can't engulf it, you can't find it, and so it's hard to get rid of. And there are other steps along the way where different kinds of microbes will try to fight this process in order to survive. Some of them will escape from the phagolysosome, right? So some of them prevent, there's a whole bunch of different things they can do. Oh, and then one last thing I want to say, because I think it's kind of cool, is that this lysosome, which contains these digestive enzymes, one of the things that is actually made that's going to destroy the contents of the phagolysosome is toxic reactive oxygen species. And we've talked about those in the context of aerobic respiration, right? You get to the end of the electron transport chain, you dump your electrons on oxygen, and you can sometimes create these toxic oxygen species that are dangerous, and we have to neutralize them, either with using catalase or superoxide dismutase. But in the lysosome, we make them on purpose to try to kill microbes. All right, so I told you that there are ways that some pathogens have developed in order to evade this process of phagocytosis. So some capsulated bacteria have an M protein, which I mentioned in chapter 15. Remember, and I warned you I would come back to that. So that M protein in the capsule actually prevents them from adhering to phagocytes, so they don't get taken up in the first place. And Streptococcus pyogenes and Streptococcus pneumoniae are two examples of that. Now, there are others that actually can be engulfed, but they will kill the phagocyte. And that includes Staphylococcus aureus strains that make toxins, exotoxins called leukocidins, leukocidin for white blood cell killer, right? Listeria, has anyone heard of this? All right, this is something you try to avoid getting when you're pregnant, so you don't want to eat certain kinds of processed meats because you can have listeria. And what they do is they use various methods to actually explode the phagocyte. So they attack the phagocyte and make holes in it. I told you there were some that could actually escape, right? So if they got engulfed and they ended up in this compartment, they actually are able to get out of it. So how do they do that? The ones that can do that include Shigella and Rickettsia. Shigella, of course, being this toxin-producing pathogen that E. coli has received the toxin from that one by a viral infection. So both of these have the ability to escape phagosomes. This one does it by reorganizing the actin cytoskeleton. And we talked about that in chapter 15. Some of them prevent the phagosome and the lysosome from fusing. So if we go back to this picture, you engulf the microbe making a phagosome, but this is a relatively benign compartment. You're just kind of stuck there, but you're not being killed, right? It's like you're in prison but not on death row, which is a really terrible analogy. But so some of them can do things that will prevent this next step. So they'll just end up floating around in this phagosome, but they're not dead. And that includes HIV and Mycobacterium tuberculosis. They prevent that step from happening. And some of them can survive in the phagolysosome. That includes Coxiella burnetii, which we haven't really talked about. All right, so that's all different ways that we can evade phagocytosis. How do phagocytes get to the sites they need to in the first place is actually really cool, and I will try to remember to post this video. I might show it in class if we have time, but white blood cells are typically floating in your bloodstream, and if there's some sort of problem, then the tissue damage in the area of the problem will cause signals to be sent that are detected by these guys, and when they sort of hear this call, then they get all flat, and they squeeze in between the cells that line your blood vessels in order to get into the tissues and help out. And so how do they know this? How do they receive these signals? So on the surface of these white blood cells, there are receptors. They're just proteins that are sticking out of the membrane, and they'll bind to some ligand that causes them to flatten and squeeze in between the layers of your blood vessels and come into the tissue. They also are adhesion molecules that allow the cells to stick to, so, you know, white blood cells are rolling through your bloodstream kind of at high speed. So they need to have some way to slow down, grab onto this region where they're needed, and then squeeze in between the cells of the blood vessels. So that they use adhesion molecules to do that. So in an area of blood vessels near where there's some problem, the endothelial cells can actually put up little sticky fingers and grab onto white blood cells as they're passing by in order to get them to come in. There are also a bunch of signals that are just floating, that just get secreted and float through by diffusion through the fluid in your body, and that includes cytokines. They're just a generic term for signals that one cell makes to send a signal to another. And they're bound by receptors on the surface of the cell, and they can induce all kinds of changes. Okay, so this is something that cells in your body use to talk to each other all the time. But specifically, there are different kinds that have specific effects. And when it comes to immunity, these are sort of the relevant ones. So chemokines are things that induce chemotaxis. What does that mean? That means they're signals that are basically saying, come here. Like, you need to come to this site. And the site where they're needed is going to be a high concentration, so the cells are going to move in the direction of the high concentration. There are also things that a cell can receive that will induce it to multiply and differentiate. So it would take an immature white blood cell and turn it into a mature one. You will have to remember this, and we'll talk about it more in a little while, but interferons, there's 3 kinds, and they're used primarily to send up signals about a viral infection. There are interleukins, so again, leuc meaning white blood cell. So interleukins would be signals that the white blood cells send back and forth to each other. And this is another place where both the innate and the adaptive immune response are talking to each other. And tumor necrosis factor, which is a signal that usually is telling cells to die.